charges. Now, this is a simple case. Well, one variant has one pathogenicity assertion, another the other, but the other was evaluated later. Here is the contradiction, right? These are one is this is the contradiction of pathogenicity assertions, and this is difference in the evidence. And because this one has stronger evidence that was evaluated later, you may try to probably trust this one. You also can see the link toward the amino acid change. They call the same amino acid change. You can have a link to their allele frequencies. So in the context of all this information, you can actually figure out which one to trust. A more complex case where we have actually three variants that have one pathogenicity assertions and one variant the other, and this and that variant have a single piece of evidence. This one is supporting evidence. Moreover, there are three of them, right? So we probably trust this. But of course, you can examine links to the frequencies and other supporting information before evaluating this one. So in terms of mouse links to mouse variants, I think the link toward homologous amino acid change in mouse and human, that link is to be established, as well as something equivalent to the mouse database linking nucleotide amino acid variants, so that these kinds of types of information can be collated and used for inference. One important bit is, again, adding new layers of data, and we actually kind of ate our own dog food, as the computer engineers say. We try to use our own project to actually test this concept of layering information on top of the existing information using our allelic epigenome project. So this is as part of the data analysis coordination for the epigenome project. It started 10 years ago. Just 1 month ago, actually, we published the final paper where we linked genetic and epigenetic variation by looking at allelic imbalances due to regulatory variation in the human genome. Using information from 13 donors, about 50 whole genome bisulfate sequencing experiments involving 27 tissues, 9 cell types. So the key element was really whole genome bisulfate sequencing and looking at allelic imbalances that may be caused by variants in cis. Each methylation of each CpG on every reads was assessed in the context of the specific allele, and special focus was on the read spanning heterozygous loci, where the heterozygous loci are either markers or actually causative variants causing the change in methylation in cis. It turned out actually thousands of transcription factor binding loci actually have these kinds of allelic imbalance patterns, and the effects in fact are mediated by transcription factor binding affinities to different alleles. So also we developed a sensitivity map of the genome in terms of sensitivity of regulatory elements to genetic variation as estimated based on frequencies of these imbalances at heterozygous loci. So the big message is CpG island and CpG-rich promoters are buffered against change. So allelic imbalances are less likely to occur than the background, whereas CpG-poor promoters and enhancers particularly are highly sensitive to allelic variation, where allelic imbalances are highly likely to occur. So the most actually unexpected pattern was stochasticity. So what we mean by that is that there's no gradual difference. Even though percent methylation is fractional between the two alleles, there's no intermediate level of methylation. It's either on or off. The difference is a fraction of on or off states, which means that the alleles actually modulate the frequency of transcription factor being the bound state of the transcription factor, with some factors preferring methylation, some preferring non-methylation, and actually this is true for a majority of hundreds of transcription factors we looked into. So the message of this allelic epigenome map is that about 5% of the epigenome shows absolute allelic imbalance, which means beta values of at least 30%. These imbalances associated with allelic transcription in cis, so these are marks of gene regulation. And we have about 200,000 such loci with these allelic imbalances, thousands of them regulatory loci, and each individual actually harbors at least 200 variants that are in purified selection and show these allelic imbalances, which means that these variants do have impact, presumably, on human health. So how did we integrate this? Here is how these user-contributed links show up in the allele registry along with these other links. And if you click on it, you get some machine-readable content in JSON-LD. You probably will not use it, but your programmers will be happy because they can serialize it as a graph. They follow the latest standard of node representation, right? They can be understood by Google. And they contain information about transcription factor binding, tissue